Hi, Dinky.
Hi, Emma. And hello, everyone. We'll wait for another couple of minutes since still people are joining.
Should I do a screen sharing test?
Can you share the screen?
Yeah, I think I need, it says the host disabled participant screen sharing. So if you can.
Okay, now you should be able to.
Yes. And here. And you see the right screen, right? I do like this.
Yes, we can say.
I will wait a second.
Okay, so maybe we can get started now. So hello, everyone. Welcome back to our online seminar series on single-cell genomics. Today, it's the third week, and for the past 2 weeks, we have been looking at what is the human cell atlas and what are the different sequencing technologies available for single-cell sequencing. So today, also, we have a very exciting talk from Ms. Emma Dann. So Emma is a PhD student from the Teichmann lab at Wellcome Sanger Institute. She is supervised by Dr. Sarah Teichmann and Dr. John Marioni, and she has been working on developing new statistical methods for comparative analysis of single-cell genomics data. So thank you, Emma, for joining with us here today to talk about the computational side of things in the field. So without further ado, let's hear from Emma.
Thank you, Deniti, and thanks for inviting me and for organizing this seminar series. I hope you can see my screen properly. Please shout, Deniti, because I no longer see you.
Yes, we can
Yes,
see.
Cool. Okay, so today I will give a bit of a sort of fast but hopefully semi-comprehensive overview of essentially the fundamental methods for analysis of single-cell RNA sequencing data. And I know that in the previous weeks, you also discussed other types of technologies such as multi-omic and spatial. I won't touch much about these because you know, a lot of the basics that are used for single-cell RNA-seq analysis also translate to those type of technologies, and I will give you resources at the end. But I think it's really important to start from essentially the basics and what are the first fundamental steps and that we need to do when we go from having an experiment to having our data sets. And the key essentially abstraction that makes single-cell RNA sequencing so powerful is this idea that we can consider a cell. A cell has, you know, a lot of different characteristics, but we can consider it as essentially a bag of RNA, and essentially the collection of mRNA molecules and from which genes they come from is actually very informative about what a cell is doing and how different it is from all the other cells in a tissue sample. And so essentially starting from data that essentially looks like this, where for each cell we count how many times do we see a set of genes, and for the human genome this will be around 20,000 genes. We are interested in using these matrices to essentially understand structure in the tissue, which cell types are there and what are they doing. And today I will be talking about three main essentially parts or essentially analysis that are routinely done, I would say, in single-cell analysis research. And I will talk a bit about pre-processing. So how do we remove low-quality cells and clean up that original count matrix? Then I will talk a lot about how do we identify cellular structure, and the type of questions that we ask when we start doing this is essentially asking which cell types are present in my sample. And also looking at processes such as differentiation trajectories, and this is what we'll talk about. And then I will also touch upon integration across samples. Why do we need to integrate data from multiple samples? And the main questions regarding this part are really looking at which cell types and states are shared across samples and which are different. Are there condition-specific differences? So let's jump right in and start with the pre-processing part. So as I said, when we get essentially our the rawest type of data that computational people like me tend to work with in single-cell RNA-seq is this count matrix. And ideally, we would, if you remember from the experimental lecture, essentially what we'd hope that each of these columns represent is one of these droplets. In most of the data that we use comes from these droplet-based technology, where in each droplet we have a single cell which has a collection of mRNA molecules that are captured by the barcode read. But in practice, in the original data matrices, a lot of the columns actually represent different types of artifacts that we might have coming that might come from essentially the experimental procedure, and we have to essentially remove out those types of artifacts. So there are different kinds of artifacts, and here I've listed a set of tools. These are computational packages. If you Google them or look at them up on GitHub, you can find them, that essentially deal with all these different artifacts. But essentially the types of problems that we might have is, first of all, we will have a lot of empty droplets. A lot of the basic single-cell pre-processing pipelines already incorporate essentially an elimination step to remove out these empty droplets. But essentially, in this case, we will see that the sum of the counts for a specific cells will be very low, and there's others essentially different ways to figure it out. But first of all, we have to remove all these empty droplets. Then we might have RNA that is not coming really from the cells, but it's only found essentially in the in the reagent mixture for each droplet. And this usually comes from cells that break during the process and release their RNA inside the reagents in what we sometimes call soup. So there's several methods to identify if there are mRNA molecules that basically don't match what we would expect from the profile of a given cell. Third artifact is having doublets. Essentially, you could have the two cells by mistake end up in the same droplet, and this happens if you are essentially overloading your experiment. And again, here you will observe a much higher number of mRNA molecules than expected, and there's different ways to tell apart whether you have essentially a combo of cells, and these come up, and these are actually quite hard to detect, but there's several methods that I've tried, but it's still quite a sort of a frequent type of artifact that you see very frequently. And then you might also have cells that are simply essentially dying, maybe because of the dissociation process. And one of the ways in which these are told apart is by looking at the percentage of reads that come from the mitochondria, which essentially indicate that the membranes of the cells have been broken. So the first step is in most analysis is really to try to take out data points that actually represent all of these artifacts to just keep our very clean looking cells. A second step in pre-processing that is done by really most analysis is applying some sort of normalizing transformations. So the purpose of doing normalizing transformation is twofold. First of all, we essentially divide the counts for each gene in a cell by the total counts in a cell. And this accounts for the fact that different cells might have been sequenced at different depths. So you might have a larger number of mRNA molecules in certain cells just because there the PCR reaction just worked better. And so the first normalizing sort of step is to divide by this total counts of cells. And then we apply a log transformation. In particular, usually the transformation is where we sort of add a pseudo count to the normalized counts and then apply a log. The pseudo count is used just because otherwise we would have some weird output if we have zeros, and we have a lot of zeros in these data sets usually. So this is why we add a pseudo count. The reason to apply this log transformation is actually that we want to make the variance in our data less dependent on the mean. So in the normal counts, what you see is that the genes that have a higher mean, so that are just highly expressed in general, will tend to have a much higher variance between different cells. And this is true even after you've applied the normalizing transformation to the dividing by the sum of the counts. But when you apply the log, you tend to minimize. It's not perfect, as you will see, but you tend to sort of reduce the dependency between the mean and the variance. And the variance is very important when we essentially try to start looking at which genes are highly variable, which genes distinguish cell from each other. And so we don't want to essentially all the differences between cells to be explained by a set of very few highly expressed genes. And, this might seem like a simple transformation. Actually, there's been a lot of research in general on how you should normalize single-cell RNA sequencing data. And I think a very good paper to learn about this is this one that I've put here, which actually compared a lot of these different methods and found that this very simple strategy is actually super effective for most of the downstream tasks that we are interested in. But if you want to go deep into this topic, I would really recommend this paper here. At this point, we have essentially a usable data matrix where we've removed some artifacts, removed some technical differences between cells, and we can start looking at the fun questions. So how do we start identifying which groups of cells are present in my sample? And the main sort of point of this analysis is to do some sort of dimensionality reduction. So the problem with our original data matrix, it's not really obvious in this illustration, but the problem with our original data matrix is that essentially this is a very high dimensional space. So we're looking at a space of, for the human genome, this will be about 20,000 genes. But in practice, there is a lot of redundancy in these genes. So a lot of genes have actually, by and large, the same expression expression profile across cells. And we can think that actually the gene expression space, in a way, we imagine that it's actually much smaller than the space of 20,000 genes. If we consider the gene expression space to be the space that separates cell types from each other, we would expect to have maybe in the tens of cell types. So we want to essentially find a good approximation of this gene expression space where two cells that are more similar to each other are more likely to be cells of the same type. And to measure these cell-cell similarity, doing this in a very high-dimensional space has a lot of problems. It's very hard to tell apart data points on a very high-dimensional space compared to how easy it is on a very low-dimensional space. So when we start trying to look for cell type identities in which cells are closer to each other, we need to do dimensionality reduction. And in practice, there's many, many different ways to do this. And again, it's a very large field, but I would say that there are three key types of analysis that apply some sort of dimensionality reduction. The first is the idea of selecting highly variable genes. So we want to retain only genes that are informative to separate cells. A lot of genes will be just expressed everywhere and they don't really help us in distinguishing different cell types, while we expect to have some set of genes that have a variance in gene expression that is much higher than what we would expect from the mean. So we want to retain these genes that seem to essentially be important to diversify cells. Then we apply some sort of dimensionality reduction technique. And the main technique used as a sort of first line analysis here is principal component analysis, which is very used in a lot of genomics applications. I will not go into depth about how principal component analysis works. But this is only, PCA I would say is like the first line of analysis, but there's also many other types of dimensionality reduction strategies that essentially go from having cells in a space of thousands of genes to a space of tens of dimensions. And once we do have this low dimensional space, this is actually a space where we can reliably quantify similarities between cells, so distances between cells. And when we have a space where we can calculate similarities, a very commonly used, again, representations for cells is the K nearest neighbor graph. So the idea here is that for each, I will build a graph where each cell is connected to K of its nearest neighbor in this low dimensional space. And K can be 10 cells, 20 cells, 30 cells. It depends really on the type of data. And K nearest neighbor graph are awesome because they capture some sort of curved arrangements of cells in gene expression space. And just to give you an idea of why nearest neighbor graphs are so useful, is that because if from essentially this connecting a cell to its local neighborhoods, we actually see structure that emerges. Here it's an example where we can see that by connecting the dots that are closer to each other, we first start seeing sort of some body part, and then we can see that the structure of a whole elephant becomes apparent. So by doing this local connection, we actually approximate the global topology, so the global sort of representation of our data set. And this is what we'd hope that it's doing also in our cells in this gene expression space that is a bit more abstract than the elephant. And they're also very efficient. Nearest neighbor graph are very efficient to compute, and they're very efficient to store also because they're sort of sparse 1-0 matrices. And they are the basis for a lot of downstream analysis that are used to identify cellular structure, namely 2D visualization, clustering, and trajectory analysis. So 2D visualization, essentially we want to, because as we said, this space of gene expression is very abstract to imagine, even if we've gone down to a few principal components and not millions of, not thousands of genes. So we frequently use these 2D projections that take this low dimensional space in this KNN graph and try to put all the data points in a space of 2D. And the most used algorithm for this is the UMAP. Again, if you want to have a more in-depth look at this, I would recommend playing with this website. Here is just showing an example where we go from having a 3D positioning of points that represents a mammoth in 3D to its 2D UMAP projection. And yeah, you can see there's a lot of distortion in these points, but by and large, we can still distinguish a lot of features of the mammoth. We can distinguish, its horns, it's legs here, it's like a body. And so again, we use these 2D UMAP representations because we can't even imagine what the real gene expression space looks like. And they are very useful. Here I'm showing an example of a simple single-cell RNA-seq dataset from blood cells. And here each point represents a cell, and points are arranged in one of these two-dimensional UMAP embeddings. And they are colored by how many molecules of this particular gene, NKG7, each cell is expressing. And NKG7 is a marker for NK cells. And even without having to do any clustering or complicated analysis here, we can already tell from the 2D UMAP visualization and plotting the expression of these genes that we probably have NK cells here, and how many are they? And so I think that, you know, UMAPs get bad rep, but they're actually very useful to explore your data for exploratory analysis of your data. Then the other main application for which the k-nearest neighbor graphs are used are clustering. So as you know, if you've done genomics in a lot of other fields, there's a lot of different algorithms that you could use for clustering. But in practice, in single-cell genomics, the most commonly used algorithms are community detection algorithms that work on this k-nearest neighbor graph. And two of the most popular one are called the Louvain algorithm and the Leiden algorithm. And the idea here is that essentially we want to separate points that are very highly connected with each other on the KNN graph. And here I'm showing, for the same datasets that I was showing before, the essentially results from clustering on this dataset with one of these community detection algorithms. And yeah, you see we have separated out with what we thought were probably the NK cells before. And clustering is very useful. The idea is that essentially clusters of cells often represent cells of the same type that have the same function. And as we've learned from analyzing many data sets, one of the most useful things you can do with the single-cell RNA-seq data sets is going from this very abstract assignment of cells to clusters that don't really tell me much about the biology, to actually start examining marker genes for your tissue of interest. In this case, we were looking at blood. And by checking which clusters express which gene, we can actually go from the abstract clustering representation to a cell type annotation, meaning that we can give a label that is interpretable, that is usable, and that essentially allows us to also compare maybe with previous research with other data sets. In this case, by looking at a set of marker genes for blood cells are very well known, and so you can sort of distinguish quite easily the different immune cell types in blood. In practice, this is a very time-consuming step for a lot of analysis tasks because there's a lot of systems for which we don't really know well the marker genes, and we might identify a cluster that actually expressed a combination of marker genes that we've never seen before. There's actually examples of single-cell RNA-seq studies that, starting from this simple clustering approach and looking at marker genes, actually have identified a lot of new or rare cell types that we didn't know existed. And you could think that this is also a classic, essentially, problem that could be solved by machine learning. And one of the, and in fact, this has been essentially something that a lot of computational research in single-cell RNA sequencing has looked at. So how can we essentially use pre-annotated data sets to do some sort of semi-automatic, automatic cell type annotation? Here I'm showing one example which came out from our group. This was a project led and still led by Chuan Hu, who is essentially, the idea here is that we take a lot of data sets that have been labeled by experts before, we harmonize these data sets together, and we use them as training data for a logistic regression model that we can then apply to unannotated data to get a first pass, essentially, prediction of which cell belongs to, essentially, which cell type is for each cell. And here I actually put semi-automatic cell type annotation because what we've learned from using these methods in several scenarios is that actually they're not, they're not perfect, and they're usually used in combination with the manual marker annotation approach, but they can give you a pretty good guess of what are the main cell types in your data sets. And cell type also has a whole database that you could look at if you're interested in using this type of data. A second application for KNN graphs and for identifying cellular structure is actually the idea that from a snapshot of cells in a given point in time, we can actually learn a dynamic process such as differentiation trajectories. And so the idea here is very similar, like, how do we do this if with a sample for which we're actually destroying the cells, we're not really seeing where a cell is differentiating to or where it's, who its progenitors were. But this is a similar idea to essentially taking a picture of, ants crawling out of the earth. By taking a picture of ants crawling, you only have one point of view, but you would still be able to tell where they're going and where they came from, right? And so the idea is that in a lot of single cell data sets, we actually see similar, essentially continuous paths in the gene expression space after we've done dimensionality reduction, etc. And we want to be able to order cells so that the cells that are more similar to each other are probably in the same trajectory of differentiation, and cells that are further away are more likely more differentiated. And again, here KNN graphs are very frequently used. And some of the most successful methods to do this pseudotime ordering of cells, we call it pseudotime because it's not real time. It's essentially giving you an idea of in which order differentiation of cells are, but it's not real time. So a lot of pseudotime algorithms actually rely on doing random walks on k-nearest neighbor graph. So if we say that we have identified with marker genes a cell that is a stem cell or a progenitor cells, if we start essentially walking along the graph from these cells, where are we likely to end up? And these type of average of random walks is frequently used as a method to identify, to order cells in pseudotime that usually frequently represent differentiation trajectories. As a final sort of analysis task that I want to talk about, which is becoming more and more routine because we are collecting more and more samples from this type of data, is talking about how do we do multi-sample single-cell analysis. So the reason to do multi-sample single-cell analysis are very practical. And even, one of the main reasons why people started doing this is simply to increase the coverage of rare cell states. If you only take one tissue sample, you're not likely to capture cell types that are very rare. Then we want to essentially have multiple cells from multiple donors or samples to essentially assess, okay, is this type of cell that we're seeing an artifact of our sample, or is it found consistently across samples? And then we want to be able to quantify differences if we have samples that come from different conditions, for example, samples from a healthy patient and samples from a diseased patient. The problem with working with a lot of samples together is that there's often batch effects. And what I mean by batch effects is that essentially cells that come from the same sample that have been profiled together are more similar to each other than cells of the same type from another sample. And the problem with batch effects in single cell analysis is that they are often not the same for different cell types. So you might have a strong batch effect in a certain cell type and a smaller batch effect in another cell type. So it's kind of hard to just regress this out with a simple linear model as you might do for bulk RNA-seq analysis. And so there's actually a very active area of single cell RNA-seq analysis that has been focused on this task of data integration, with essentially tries to minimize the batch effects and essentially find a latent space where the differences between batches are minimized. And this, again, there's a million methods to do this. Some are based on variations of linear models. Some are based on deep learning models. This is a paper that I would recommend if you want to learn about what are the similarities and differences between these approaches, where they actually benchmarked a lot of these different approaches. Here you see only a subset of the methods that they benchmark in this process. What I want you to sort of take on from this slide is that whatever method you use, there's always going to be a bit of a trade-off between batch correction, so minimizing the differences between batch, and conserving the biology. And in this case, what we mean by conserving the biology is, for example, conserving those differences between cell types, because you could just essentially remove all the information in your data set and you would have perfect mixing between batches, but arguably that's not very useful. And so when considering how to do data integration, there has to be sort of an analysis of this trade-off. How much information are you losing from essentially minimizing the differences between your samples. If you do have, once you do have sort of successful integration, and that paper also proposes a lot of different metrics that you could use to assess whether your cells are well mixed. Frequently in these multi-sample experiments, we actually have multiple conditions. So for example, here I'm showing a dummy example where we might have cells from 3 different cell types, and a subset of these cells come from control patients, and some cells come from samples that have been treated with some sort of drug. And so the main there's essentially 2 main parts to comparative analysis of single cell genomics. There's many more, but I would say that these are the 2 most commonly common strategies. One is essentially assessing if there's differential cell abundance. So is there a significant difference in proportion of a given cell type between the control and the condition? And then differential gene expression. So if we take each of the cell types, do we see that a certain gene is more highly expressed in the cells of that cell type that come from the treatment compared to those that come from the control? And, differential cell abundance and differential gene expression can essentially identify the same phenomena. It depends a lot on how you clustered your cells, at what resolution you clustered your cells. One important point that I wanted to take away from how you think about comparative analysis in single cell genomics is that here the grouping is not essentially the units that we use for comparative analysis are no longer cells. As you can see here, we're actually grouping cells by clusters, cell type clusters, and samples. So frequently when we do differential gene expression analysis, we don't just test the differences considering cells as replicates, but we actually aggregate gene expression profiles that come from cells of the same sample and of the same cell type. And this has been shown in several scenarios to essentially reduce a lot the false positive rate that you get from these experiments. Comparative analysis is still a very active field of research, so there's no sort of strong best practices. It's really still a bit of the wild west, but I think it's an interesting new avenue that we're starting to look at now that we have more and more samples from different conditions. As a final sort of example of very single cell specific analysis that you can do with this data that you really couldn't do with bulk RNA sequencing or other sequencing technologies, I just wanted to mention cell-cell interaction analysis. So one of the fun things that you can look at when you have data from multiple cell types is you can check whether by looking at having the data sets of ligand and receptor that act between cell types to sort of for close short range interactions, we can check whether a certain cell type is always expressing the receptor and another cell type is always expressing the ligand, and that gives us a pretty good idea that probably these 2 cells are interacting. And our lab has worked on this historically, and one of the first tools that came out to do cell-cell interaction analysis was this cell phone DB, which is essentially a database of these ligand and receptors. And by simply doing a permutation test, we check whether there are certain cell types that are more frequently expressing ligand and receptor together than all the other cell types. And this can give us an idea that cell type A and cell type B are working together. There's a lot more that I didn't have time to cover here, but I wanted to give you, and I've told you about many different methods, but you might be like, oh, well, thanks. How do I actually do this if I do have data and I do want to learn more about this? So here I've got a list of resources that you could use that have sort of in-depth tutorials with code, both in Python and in R, to learn more about single-cell analysis and many more steps that I haven't covered here, including analysis of multi-omic, analysis of spatial data, and also some databases that you could use to take out data sets if you're interested in doing these analyses. There's really a lot of publicly available data sets that you could work with. And with that, here are my contacts. So if you have any other questions that come up after the lecture, please feel free to contact me. And thank you so much for listening. I'll be happy to take any questions you might have.
Thank you very much, Emma. That is a very nicely described presentation. You gave us an overview of different stages from early to the last downstream analysis stages of single cell analysis, and how the different data mining and machine learning techniques can be applied for different types of questions arising. So is there any question from the audience for Emma? If you want, you can also type your question into the chat box. Maybe I will, oh yeah. So we have a question. So Bhagya is saying, it's really interesting, thank you. In data clustering, are these 3 steps have to follow as twice one after other, or we can select as required? Can't we use t-SNE or NMF? PCA data loss is higher, bulk cell transcriptomic is it same in single cell?
So thank you for the question. That's actually quite a good point. In some of the early days of single cell genomics, people were actually, for example, doing things like TSNE or UMAP and then clustering on that space. But this has been shown to actually introduce a lot of artifacts, and essentially, that there is this notion that the space, the UMAP space, is good for visualization of clustering results or of a low dimensional space, but it actually warps the similarities between cells, especially on the long range. So you'd have to essentially tune your clustering parameters in a way that we're not really that we're not really good at doing. While if you do the same, so in a way, having that as a separate visualization steps is very important. As for example, NMF, there's a lot of methods that also use things like NMF. The idea is that in this step, using something that is very high compression like PCA is actually quite advantageous. In practice, when you do have multi-sample analysis to do, you would use an alternative dimensionality reduction method that can account for differences between samples as well. A lot of them are based on neural networks, or for example, there's some sort of PCAs that also incorporate the covariates from which batch does each cell come from. So standard PCA is more like really the bread and butter, so what you'd start with first, or if you're analyzing a single sample, but there are a lot more sophisticated methods, some of which are also based on NMF. The step of feature selection, I would say, is always the first, and it's there's some methods claim that you can go without doing feature selection, but in practice is one of the things that improves your clustering results the most. So that's always essentially done upstream. And you might not need to do a very stringent feature selection, so you don't need to go from like 20,000 genes to 1,000 genes, but it's usually more going from 20,000 genes to maybe 5,000 or 10,000. So you reduce essentially the space by half, but you still have a very high sort of high dimensional space to feed your PCA or your dimensionality reduction. I hope that answers your question.
Any other questions? Maybe I'll ask one question. So there are lots of different data mining and machine learning tools and computational tools for single cell data analysis. And do you have any advice for someone who is just getting started, let's say for a bioinformatics project, what sort of topics that they could look at specifically where there are certain gaps of the tool development or research gaps in those tools?
Right. So things that have not been where there's no best practices or so I would say, like, very biased because this is also what I work on, but I think that on the comparative analysis, so how do you actually quantify differences between data sets, and the statistics to do that robustly is very, I would say, very active area of research, and I think there's a lot of unanswered questions there. And yeah, I know also Diniti works on this, so I think we can can agree that there's a lot of like interesting unanswered questions there. And also, I think that could have a lot of impact because in practice, there's only so many new cell types that we can discover just looking at, normal samples, but actually looking at, once we have a fairly good idea of which cell types are in a lot of different tissues, looking at how these change up in different conditions, I think is one of the most interesting questions. And then I would say that another very, still, there's a lot of things to try and test is how you analyze single-cell RNA-seq data and data from multi-omic experiments. So from different molecular modalities for which you have single-cell information, how much does this differ from the information that you could get from the bag of RNA view that you get with single-cell RNA-seq? I think is still essentially to be determined. And very, as new technologies come out, there's always some new, the analysis has to follow. So when some new molecular profiling technology comes out, some computational person has to figure out how do you actually analyze this new type of data. And I think that's one of the things that is most exciting about single-cell genomics. So the technology is always like one step ahead of the computational analysis. Yeah, I hope there's a way to share the slides, or if this will be on YouTube, all the for essentially getting started, the set of tutorials that I looked up go very in depth from very like the basics to the very advanced topics. And I think those are very good because there's also like example data sets and example code that you could play with. So I think they're good resources to learn more about also what the problems are, because there's these advanced tutorials that are like, oh, this is how we do it for now, but this doesn't work because of this and that. So you can like figure out how to, there's areas that are more benchmarked and solid than others.
Great. Thank you so much, Emma. Any last minute questions before we end the session? If not, yeah, let us thank Emma again. Thank you so much, Emma, for giving us such a nice presentation. And I also would like to note the analogy that you show from this elephant view from UMapp. It was such a nice overview. Just wanted to yeah, I quite like it.
Thanks.
Yeah. Okay. Thank you so much, everyone, for joining us.
Bye, everyone. Please feel free to contact me with how the questions come in mind as you go on with your day. Bye, everyone.
Bye. See you next week, everyone.
See you.